Hi everyone, this is Ghalib, and with me we have Substantia Nigra. And today we are going to discuss a very popular video that has about 3.6 million views on YouTube. And this video is considered by many creationists or the people who deny evolution as one of the strongest videos that is against uh, evolution on the internet or on YouTube. So, in this video, there are three gentlemen who discuss why evolution is wrong and why, and at least two of them try to, one of them openly says that intelligent design is the more convincing answer to the mystery of life. And the one other, he says that uh, he aligns with him. And the third one, though he is their ally, also gives them away. So we welcome you all in this live stream. In this live stream, we are going to discuss many things about evolution. We are going to show you a lot of evidence. We are going to tell you how their arguments are flawed and why they haven't considered the whole picture, the whole modern picture, the 21st century picture of natural evolution through natural selection. And they've just jumped on the conclusion that evolution is wrong without looking carefully at the evidence and without rationally analyzing how evolution is how evolution happens and what are the mechanisms behind it. So, Substantia, welcome. And I would like to know your opinion about the topic that we have picked today and why it's important and why it's important to debunk this video that we are talking about right now.
Thank you very much, Ghalib. Hello, everyone. So, today's topic is to discuss this video, which, as Ghalib just put it, has been uh, has garnered such a huge viewership in millions. So, it has got three participants. So, the three main participants who the anchor is interviewing. So, I'll just introduce the three main participants. So, one of them is David Gelernter, who is a computer scientist and a professor of computer science at Yale. Second person is David Berlinski, who is a mathematician and philosopher and
So, this
works.
is David Gelernter, and uh, this is Berlinski on the right side, and
Yeah,
this is Stephen C. Meyer.
yeah. This is
So,
Steve Meyer, who has got his PhD in history and philosophy of science.
yeah, and
And
he has written some books against evolution, and I've unfortunately read two of them about 3 4 years ago.
yeah, so Ghalib has read the book that is the focus.
Yeah,
That's kind of the focus of this talk. I won't say focus, but that was the trigger of this video. So just to give you a backdrop, so the professor of computer science, this guy David Gelernter, he wrote a book review at uh, this website, which is the Claremont Review of Books, and the title of that book review was "Giving Up Darwin." And what he mentioned in that book review was that he was really inspired by Stephen Meyer's book with the title Darwin's Doubt. And he said, and we will show you the excerpts from the video as well. And he actually said that he was quite inspired after reading Stephen Meyer's book Darwin's Doubt. And after that, he decided that he's going to give up on Darwinism, give up on natural selection, and give up on evolution. And prior to that, he said he used to believe that evolution was true. So apparently, as seems the case, is that David Gelernter was inspired by Stephen Meyer's book Darwin's Doubt. One important thing is, as I always mention, that we by no means ever imply that we are fond of arguments from authority. And we want that if anybody has got requisite enough knowledge and can make sense and is willing to engage in dialogue and is willing to learn and explain things to people, we are fine in terms of talking and exchanging ideas with those people. However, one should remember that all of these folks are talking about molecular biology, modern biology, genetics, and its implications on evolution. None of them happens to be a biologist. Uh, this doesn't disqualify them from talking about it, but just to put the record straight, none of them is a biologist. Stephen Meyer is PhD in history and philosophy of science. As I mentioned before, David Gelernter is a computer scientist, and David Berlinski is a mathematician and philosopher. But all of them seem to be very highly educated and qualified and successful people. So we will listen to them regardless, and we will just address their points that they are raising rather than saying that, okay, they are not qualified to talk about that. I would say I would go ahead with this assumption that they are more than qualified to talk about the topic, and we will take their arguments on their merit, solely on the merit of the arguments. So the nutshell here is this: that most of them seems like that it seems like that they are trying to underestimate the sources of variation during biological evolution. Specifically, seems like that they want to restrict those sources of variations only to genetics, and within genetics also only to point mutations. This restriction that they are putting, and of course this restriction that they are putting on their discussion, is actually against empirical evidence, because the empirical evidence tells us that there are so many other sources of variation. Okay, but there, like, which go beyond genetics. Within genetics, there are like a slew of sources of variations that go beyond point mutations. We will talk about them one by one today, like in threadbare, like in total detail. We will talk about all of those sources, and we will also talk about some of the latest findings that have been accumulating over the last 2-3 decades from emerging fields of epigenetics, which is like a parallel way of multi-generational inheritance of traits in throughout life and in animal kingdom and in plant kingdom. We will talk about that as a significant player and a force in the evolutionary history of life, and other sources of variation, like the inheritance of the microbiome and the symbionts. We will talk about all these things. So our humble submission at the end would be that if you take into account all these potential sources of variation, then the evolutionary framework is very much consistent and is backed up by evidence. Okay, and in our humble opinion, the point that these three gentlemen are missing is that they think they are stuck in purely in genetics and also only within the framework of point mutations. So that's the deal. Also, one more thing, as Ghalib said, that one of them happens to be a staunch proponent of intelligent design, which is Stephen Meyer. Other two do not
Dropping it.
actually, yeah, the other two do not subscribe to, even after the end
Yeah.
of the video, they do not subscribe to intelligent design. They're skeptical of evolution. Okay, but they do not consider that intelligent design
Intelligent design is an answer. They don't.
is a viable solution to it.
Yeah.
So, with this backdrop, what we're going to do is maybe Ghalib will play a short clip, uh, which is kind of their main, apparently strongest argument, because they frame it in the language of mathematics. So, and the video has been titled as Mathematical Challenges to the Darwin's Theory of Evolution. So let's hear them out their strongest uh, argument, and then we will deconstruct each and every bit of that argument. And somewhere during the video, we will also present a mathematical challenge to the deniers of evolution, because we will show you results, which of course are backed by evidence as well, but we will show you results in mathematics which prove, like as in mathematical proof, which prove that evolution is a mathematical inevitability. Okay, so we will put our challenge as well, our mathematical challenge to the deniers of evolution. So stay tuned. So let's start with reviewing one of their clips in which they talk about they put forth their apparently strongest mathematical objection.
Construct the theory for that.
All right, now somebody give me some notion of the math here. Things are more complicated than Darwin knew. We understand that producing new forms of life now means not just new shapes, new activities in which life engages, but a prior code. Or is that fair? You're the man who knows code.
You know, the mathematical element of this, not of population genetics in the complex, sophisticated, predictive sense that Steve was referring to, but just the simple issue of the code. It is remarkable for young people to learn in high school. It's remarkable for me or in elementary school to learn that proteins, molecules are assembled because there are codes. There are codes in the nucleus of cells that spell them out, character by character, codon by codon. This codon means this amino acid, and the next one means that, and the next one means that. But the mathematics underlying these codons is very simple, and Darwin could perfectly well have understood if he had the facts. Each one of these positions has to be occupied by one of 20 amino acids. Okay, so you pick one of 20 guys for this position and one of 20 guys for this position.
You talk about visualizing a string of beads.
Yeah, like a
Okay.
string of
As
beads.
you're building a protein. Yeah.
So you have four
building a
different colored
protein.
beads, roughly?
I'm building a protein out of amino acids.
Yes,
And I'm doing it by choosing the amino acids one by one by one by one by one.
yes.
And I have 20 choices each time. Now, if there are several hundred of these things in the string, in the bead, in the necklace, it's a big necklace that wraps around your neck 18 times. So there are several hundred, or 5 times, whatever it is. That's a huge number of possible choices. The number of ways in which you can arrange the emerald followed by the ruby, followed by the opal, followed by the chunk of platinum, and another ruby, and a diamond, and a aquamarine. The number of ways you can arrange that is huge, grows exponentially as the string gets longer. So even when the string is short, even if it's a cheap necklace for your very first girlfriend, and it's all you can afford, it's still, there's an astronomical number of choices. And Darwin could easily have computed that. He just didn't know about the amino acids. He didn't know about the necklace. He didn't know about the string. It's not the mathematics that stumped him. It's the biology. The mathematics is simple. A high school student can compute how many choices there are if there are 20 gems for position number 1 and 20 gems for position number 2, and you have 60 gems altogether.
And the task here, let me,
It's 20 to the 60.
try to mute, so I'm quoting from your,
Even you can.
Even I got it.
you got it right? I'm
Even mathematicians can understand it.
David,
Yes.
Berlinski has a
So I think this is the portion that we wanted to show. And basically what he's trying to emphasize here is that it's impossible. Darwin never understood it. Secondly, the mathematics behind the genetics is very simple. Genetics is just like a code. The DNA is just like a code of any programming language. And that's how he's trying to put it here. And he thinks that there are a lot of combinations that could be made from these 20 amino acids, but somewhere they don't believe that through mutations, which they ironically believe that the only mutations that happen are point mutations, and we are going to show you that that is not the case, and these people deliberately overlooked it, or I don't know why they didn't perceive mutation as it should be perceived, or according to the 21st century understanding of microbiology and molecular biology, we know that uh, the capabilities of mutation within organisms is huge, and we will explain all of that to you. We will show you how there are a lot of mechanisms within living organisms through which the genetic code inside a living organism can mutate. And it also depends on the different types of organisms, because some organisms have um, have the genes that can have very short lifetimes, and their genes can replicate within a very small time, uh, you know, too many times, and there are many more chances of mutation in those organisms. And there are organisms like us whose cells divide and multiply only at the beginning when they are planted in their mother's womb, and that's the time when there is a possibility of our genes changing. But for microorganisms it's a different story. So now we would go to this uh, slide that we have from what they showed during this um, all of their discussion. So this slide I would
Yeah, while you bring up the slide,
yeah.
so guys, you have heard this objection, and I will repeat this objection shortly. So if you could go to slide number 2.
Slide number 2. I think this is slide number. This is slide number 2. Yeah.
Yeah. So just for the sake of completeness, so this guy, David Gelernter, seems extremely inspired by. So the slide just disappeared.
Yeah. I'm going to just. The scroll on my mouse is kind of really crazy because you just touch it once and it just goes bonkers. So that is what happened. Okay. Yeah.
Yeah. So this is just for record purposes, that this is David Gelernter, who is writing in his review that Stephen Meyer's thoughtful and meticulous book Darwin's Doubt convinced me that Darwin has failed. Okay. So, and the other person also somehow chimes in, David Berlinski over here. So he is convinced after reading this book. So let me just summarize what he has said. He has actually said that the probability of building a useful protein is extremely small. Okay, this is essentially what he's argued for. So again, Ghalib, you can.
And unfortunately, this is one of the books that I have read from Stephen Meyer when I was preparing for a debate with, one of the famous Pakistani biologists. At that time, I read one, two of his books, and I think 2 books of Michael Behe as well. So yeah.
Yeah. So Ghalib, you can actually bring up your stuff where you wanted to explain different sources of variations within the genetic framework and what is the potential of producing new and new variations within the genetic framework. In the meantime, I will just, for the sake of completeness, I will just explain some very basics of molecular biology so that everyone from the audience can appreciate what Gelernter is saying. So I think our audience is informed enough to know that our DNA is composed of string of nucleotide bases. Everyone knows those are ATCG, adenine, thymine, guanine, and cytosine. And the deal is that from these bases, the first step, which is referred to as the transcription step, that transcription step actually creates a single-strand RNA. So DNA is a double-stranded nucleic acid, and if you focus on the bottom figure over here, the transcription step is referred to the step where DNA is converted into a single-stranded messenger RNA. And then the messenger RNA is converted into amino acids to make things extremely simple. These 3 digits or 3 characters that you see, they are referred to as codon. And this step of going from mRNA to amino acid is referred to as translation. So in a nutshell, from DNA to the synthesis of a sequence of amino acids or proteins. Protein is just a sequence of amino acids. Then this step involves 2 steps, which is first transcription to RNA and then translation. Pretty much basic stuff. And we will get into more details.
So,
yeah, go ahead.
So basically what you are seeing here, as you see on the top here, you can see that 3 of these different nucleotide bases are grouped together, and this is known as a codon. So here AUG is basically a codon that is encoding a protein. I don't know what protein that is, but it's known as MET. Maybe it's a long form, a short form for some other protein.
Yeah, these are
amino acid. So when the amino acids join, this is a chain that they were referring to, and that is basically a protein. So usually the proteins curl up and form a certain kind of shape that performs a certain type of function in cells of a living organism. So this is what is basically happening. So when they say that here adenine, uracil, and guanine, basically because this is mRNA, this is not DNA. So uracil is basically the one that is here. So it's basically coding a protein. Amino acid, and that amino acids are joining up and making a protein here. So we will go back to the first slide here, and
one thing. So of course, this string of amino acids is what is a protein, and as Ghalib just mentioned, that once the amino acids are strung in one long, like a string, the protein actually conforms to a 3D shape or folds to a 3D shape. I'm sure everyone must have heard the problem of protein folding, and the AlphaFold in 2020, and one of the Nobel Prizes of this year went to people who developed AlphaFold as well, which was a machine learning based algorithm to predict the structure of proteins from a sequence of amino acids. So as it turns out, the 3D structure of the proteins actually determines its properties. One of the major functions, one of the most common functions of proteins is to serve as enzymes or to facilitate reactions. So the 3D structure of the protein actually makes sure that in a nutshell, the reactants of a reaction can actually align or orient in 3D accordingly, and it facilitates to bring the reactants to a proper orientation or alignment. So that's how it acts. Its 3D structure is really important. So Ghalib, you wanted to discuss this redundancy of the code, or the idea of synonymous codons, because they think that in order to construct a protein, you need to have exactly the same sequence down to the last character.
This is what Gelernter said. This is what David Gelernter said, that he said that, okay, there are only a limited number of options, and to make an amino acid from a codon, it has to be exactly like that, and he cannot be he is just totally wrong there, because as you can see, this is a chart that is basically explaining how codons are made. So this is guanine, this is uracil, this is adenine, and this is cytosine. So these are the 4 nucleotide bases. Basically here we don't have thiamine. We are talking about messenger RNA here. So RNA does not have thiamine, it has uracil for thiamine. So basically how codons are made, this is a chart that's explaining how different codons are made and how different proteins are encoded by those codons. So everything that's outside this circle is basically an amino acid. And all that is there, you can see that in the center, you can see all these 4 nucleotide bases. And all around, if you read carefully, you would see that UGAC is repeated all around. And even if you come in the outermost circle, you can see that again you have UGAC, and that's all repeating. So how is one codon being this? How does this chart explain which codons and which codons encode which protein? So you can see, let's take this top right quarter of this circle into consideration here. So in the middle, we have uracil. And when uracil joins with adenine, and then that adenine joins with a uracil again, so this is the protein that's made. But this is not the only way that this protein is made. So uracil joins with adenine, and then again it joins with cytosine, and again the same protein is made. So you can see that there are a lot of proteins that are made by a lot of different codons. Like this protein, like we can see that there are 4 individual sequences that can return, this amino acid, there are 4 different codons that result in the same amino acid. So, and this is not just 4. You can see that there are how this works here. So, and it's not only this amino acids that goes through this. You can see there are a lot of other amino acids that have 4 codons that encode the same amino acid. Then again, it's here. Then again, you can see here. So the chart, as they were saying that only a single amino acid can only a single codon can encode a specific amino acid. That's totally wrong. So Gelernter, sorry, I mispronounced his name. He was totally wrong there, because this is not the case. The case is very different. It's like, let's say if we are here in this situation where there is a DNA sequence, and this guanine and cytosine is basically inserted in middle of this here. So basically, first of all, what we have to know is that it's not just a single pair that encodes a protein. This is an amino acid. This is what we have to understand, all of us. So it's the 3 different nucleotide pairs that make a single amino acid. So AUG is basically a codon that is making this specific protein. Then again, this is ACG that's making another protein. And hence, how this is going forward, in the whole sequence, 3 bases make 1 codon, and it encodes 1 nucleotide. It encodes 1 amino acid. And this is how things go on. But let's say this was CAA, and then a C is inserted here. So then this whole thing changes altogether. How? That now this CAA, which meant something else, is replaced by CAC. And whatever protein CAE was encoding, it is replaced by this protein. So now, this amino acid. And now the whole protein from this point on changes. So this, even if there is just a point mutation, a codon changes at one point, a single codon changes, or a single base pair changes, and first 1 codon changes, but the following codons are also going to be changed. So this is known as a frame shift, because from here,
Ghalib?
the sequence is not the same anymore. Yes.
So frame shift is a very interesting idea. But before that, if you think about it, even so frame shift can create a lot of shift, as you would probably explain in a minute. But if you go to the next slide, we will come back to it. So actually the slide where you were showing the wheel and the table.
Yeah. This,
so his entire argument is actually missing the vital detail at two steps. First, it is assuming that in order to produce a particular string of amino acids, you need to have a specific codon, like the exact same codon. Okay, so he is wrong on that account. And the second thing is that he's also operating under the assumption that in order to produce a particular protein and get the corresponding 3D structure, which is actually what matters, like from the perspective of 3D protein function, he is considering that you need exactly the same amino acid. So that's why, because there are 20 amino acids, that's why in his calculation, which he thinks is very simple and simple school-going kid can understand that. So that's why he was saying that if you have to compute the probability of a particular protein, you have to multiply 1 upon 20, which means raised to the power the number of the amino acids. And no doubt, in that case, that number would be vanishingly small. So yeah, here is apparently he thinks that he is going to declare victory, and his argument is successful. However, the slide that Ghalib just showed actually shows a couple of things. First of all, these codons, which are technically the same codons from the perspective of what amino acid they correspond to, these codons are referred to as synonymous codons. Okay, so for instance, if you look at the table on the right-hand side, UGC and UGU are technically, the first row are technically synonymous codon because both of them correspond to the same amino acid, which is cysteine. And right in the middle, if you consider, so this is the amino acid, this is ILE, this is not LLE, it appears LLE, but this is capital I and LE. This amino acid is also called as isoleucine. So that's short for isoleucine. So which codons correspond to isoleucine? 3 codons, AUA, AUC, AUU. So what does that mean? That these 3 codons are synonymous codons. And there is also a concept of synonymous mutations. What does that mean? That if I'm sitting at an AUA, and if an A undergoes a mutation to C, and you end up having an AUC, nothing matters, because it would still continue to produce the same amino acid, isoleucine. So such a mutation is also referred to as either synonymous mutation or sometimes neutral mutation as well. So even if you go back to Darwin's original book, Origin of Species, he was of the opinion that some of the variations are beneficial, some are harmful, and some are just neutral, sitting there. So if there is another change, then that's called, if it goes the other way around, the third one, that is also going to be a neutral mutation or synonymous mutation. So Ghalib, before we talk about frame shift, because that is going beyond point mutation, I just want to deconstruct his second assumption as well. So I think by this time it should be clear that in order to produce the same amino acid, you don't necessarily need the same sequence of letters. It could very different sequence of codons in terms of combinations are going to be the same, because if 2 codons correspond to a particular amino acid, and if Ghalib, you could go to that slide, because there is a point that I want to illustrate.
Which slide? The first one?
The wheel and the table.
Yeah, this one.
So let's suppose you want to build a 2 amino acid long protein. This is not real. I'm just trying to explain. Let's say you want to construct, let's say, a cysteine and an isoleucine next to each other. Then the possibilities of getting cysteine and isoleucine are not so there's going to be 2 possibilities from here, and for each of those 2 possibilities, 3 possibilities for here. So there are going to be 6 possibilities for producing a sequence of cysteine and isoleucine. So he's actually underestimating the combinatorial potential in it. They are considering that, you know what, it's a combinatorial inflation, that combinatorially molecular biology places a challenge to evolution and natural selection. Actually, it is precisely because of the combinatorial explosion that we get more possibilities and combinations. So you just saw that in order to build amino acid sequence of cysteine and isoleucine, you don't necessarily need a particular set of codons. There are 6 possibilities we just see over here. So this is part 1. Part 2, he is wrong by far. And Ghalib, if you could open up the paper, which is, I think you've got some papers opened up over here. If you could open up paper number 2, I just want to show how wrong the understanding of these 3 guys is. So if you could open
This
up
is paper number 2, and I'll just try to zoom it in a little bit.
if you could
And
read,
this is
yeah. So this is a paper on the folding and evolution of multi-domain proteins. And multi-domain proteins are those proteins which are composed of building blocks of repeatable amino acids. And like where in each domain can vary independently while the other domain remains pretty much the same. I want to read a particular sentence from this actually paper. If you could scroll down, I have highlighted this. If you could scroll down.
yeah.
Okay. So I think the
Is it, should I zoom it in a little bit more?
I think that this is good. So in the second column on the right, let's focus on the green sentence. What does it say? It says often structural similarities can be present even in proteins that share little sequence similarities. So just read this. Dr. Gelernter, Dr. Berlinski, Dr. Meyer, every one of you. Yeah. I mean, this is molecular biology 101. You are saying that Darwin didn't know about molecular biology. This is molecular biology 101, which actually means that the proteins can have striking similar structure in 3D, which is actually the thing that matters on ground. And two proteins with very different sequences of amino acids can curl up into a same 3D structure. So you can consider that there are synonymous codons, there are synonymous mutations, and technically there are synonymous sequences of amino acids as far as the 3D structure is concerned. So therefore, the recommendation is, therefore, structure-based classification is more effective at identifying distant relationship between proteins that are sequence-based approaches. In any case, you see that his entire argument was based on the fact that in order to produce a particular protein, you need a particular string or a sequence of amino acids that is patently wrong based on modern understanding of molecular biology. And
so in the end, it's the structure that matters. The codons and the amino acids, and what he was saying, that there are emeralds and there are diamonds and this and that in that necklace, it does not matter. The only thing that matters is how it curls up. And there is a good chance that even if the two proteins are codonically and amino acid wise totally different, they can perform the same function pretty well.
Yeah. So what has happened is that even within the framework of point mutation, their calculation, their approach is totally wrong-footed. So doesn't make sense. Now what's going to happen is Ghalib is going to explain to us many different sources of variations which go beyond the simple idea of point mutation. Okay, so but what we have established so far, their so-called strongest mathematical argument was wrongly premised based on just surface information of molecular biology. Somebody told them that there are so many codons and so many amino acids, and the mathematicians just bounced upon it, just multiply 1 upon 20 so many times. They didn't understand the molecular biology. You have got synonymous mutations, synonymous codons, and many proteins can have the same 3D structure on the ground. even though they have got a lot of difference in terms of the sequence of amino acids. So from the perspective of point mutation, even from that perspective, their argument doesn't kick in, doesn't fly. Now, Ghalib, over to you. You talk
Yeah,
about frame shift.
if you would explain splicing and conclude this one, then we will move forward. Yeah.
So you don't want to talk about frame shift first?
Okay. You want to?
Yeah.
Okay.
Yeah,
So we
I
talked about frame shift. Let's say there is a frame shift that is happening here at basically from this point on, the proteins would be the protein would be different, and the codons are different, the amino acids would be different, but there is a chance that even then the protein could perform functional. It could be functional. It might behave a little bit differently, but it might still be, there is still a chance that it wouldn't break the function of the cell. So this is how it goes on. But the basic premise of all the argumentation that was brought forward by these three gentlemen, I think it was totally based on point mutations. And they thought that if a new protein would emerge in an organism, that was just going to emerge through point mutations, or somehow new codons would have to come together and they would result in new amino acids, and finally there would be a totally novel protein that wouldn't share any kind of behavior or wouldn't have any kind of similarity with the proteins that already exist within that organism. That's basically totally wrong. Organisms do not only modify their own or the structures, the different structures are not only modified within the organisms due to changes in codons and point mutations, and there are many other kinds of mutations. There's also a chance that different organisms give their genetic code to other organisms, and that just permanently becomes part of their genetic code. And this is how information between organisms is exchanged, and there is a greater chance of mutation and modification of genetic code. Not only that, there on a level, different kind of capabilities have been shared between different organisms because they were sharing the genetic code between them. So this is what we are going to discuss next.
With the frame shift, Ghalib, if you would like to spend just 30 seconds on frame shift just to explain. So when you start reading, for instance, from AUG, the first one, that's the correct starting point where you should start reading if you want to produce this particular amino acid. You can consider that if I have a frame shift of like 2 codons, or three codons that I start reading from the second codon ACG, well, I will just miss one amino acid and doesn't matter much. Technically, it's a long string of amino acid, doesn't matter much. So it would be pretty much the same protein. However, if you start reading from the second codon, which means you just have a frame shift of one nucleotide, base pair, one base pair, then you would start considering this as a UGA, CGG, AUC, which means that totally novel sequence of amino acids will be constructed. It could be gibberish, it could be noise, but the point that we want to illustrate that frame shift actually gives a lot of possibilities from a small change. So they say that, okay, you know what, in order to have changes, then what happens, every bit needs to change, and there need to be so many point mutations in order to get an overall mutation rate. No, as you see that only one frame shift actually changes everything. So from that point on, so that's the novelty generation potential of the frame shift mutation. It can go wrong as well, but the point is that you would end up generating brand new sequence of amino acids because of one frame shift. Okay, so carry on.
Okay, so splicing. Do you want to explain splicing?
Yeah, sure. Also, one important thing is that in the DNA, when from DNA, uh, uh, there are portions which are referred to as introns and exons.
Yeah.
Okay, so basically the coding part are exons. So the portion between two coding parts is called the intron. So what happens is that when the transcription starts, first the coding parts or the exons are excised. So that is what you might want to consider as pre-mRNA. So it's not yet mRNA. The transcription has not been completed. Okay. When these the codons, because the introns has been removed, these gaps between them are those spaces because the introns have been removed. They need to be assembled or stitched together again. So splicing is another word for stitching. Splicing means stitching. I think you are familiar with the word cutting and splicing. Cutting means cutting, but splicing means stitching or joining them together. And there is a splicing enzyme that takes care of splicing them, combining them together. So these red, yellow, and green chunks that you are seeing, these are not single nucleotides. These are chunks and segments of nucleotides. Now, the splicing enzyme can join them in multiple ways. So these are called alternatively spliced messenger RNAs through the splicing enzyme. So you see that you can get this red, yellow, and blue, or you can get red, green, and blue. As a result, what happens is that you have this complexity, a layer of complexity added to it. You can start having like these, like it's not that you are now only stuck to or restricted to single nucleotide bases. You have got the choices being made at the level of chunks of DNA, and at a bigger scale, you can have like chunks of DNA and then combined in or spliced in different ways. That actually increases the number of possibilities that you can create.
I'm going to
Right,
just share my screen and let's draw it here.
Yeah.
I've got out my tab and let's do that.
That would be very helpful. Very helpful.
Where is my fresco? Why is it not showing up? Because this thing needs to be explained properly. Okay, people. So this is what happens. Let's say we have I don't know what's happening here. Okay, so we have a sequence. This is a simple representation of a gene. So this is a gene. Sorry, I am really, bad at this right now. So what happens here is basically when maybe this is the 5' end and this is the 3' end. I am a little bit confused. Substantia, you can help me here. Does the the transcription happen from this side or that side? I'm just forgetting.
5' to 3'.
5' to 3'.
If you want to illustrate the idea, that's fine.
Okay, so the coding starts from this side. So basically, this is a gene. Let's say it's 100 base pairs, which has almost 33 basically codons in this. And let's say from here, from the start, from P' to here, we have this portion that is an intron. Intron is basically a portion that is not going to be converted into RNA. So this gene, in order for the protein to be produced by the code written in this gene, it has to first go through mRNA. So when it has to become RNA, first we discussed that uracil would replace thiamine. Secondly, the other thing is that this sequence, this interspersed sequence in between, that is not part of the coding, not the part of the coding part of this gene. So this is just going to be removed, and the final RNA is going to be much smaller than what the original gene was going to be. So genes are not like there's just only one intron in between. There,
Ghalib,
if a gene is
can you make another intron, maybe just on the right side as well?
yeah.
The
So
pla- yeah,
this is
yeah.
this is another intron here.
Let's label the white parts, the exons. Let's
Yeah.
label the
So
white
this
parts as
is E
ABC, or something
ABC.
like anything like the three white parts we have. We can label them as ABC.
So these is ABC,
No,
ABC.
A.
Yeah, yeah.
B,
Okay,
and-
okay. I got it.
yeah.
A, B, and C.
Yes.
Excuse my poor drawing. I'm doing it after a very long while. That's why.
So
So
for the same DNA sequence with alternative splicing, Ghalib, you can write what potential possibilities could be constructed.
definitely, let's say this is a gene. We say this is G1. Basically, the splicing patterns are dictated by different mechanisms in the genome. That's basically some. So let's say we have the same coding and we have the same sequence,
You can have ABC,
totally the same.
You can have ABC in the
A, B, and C.
mRNA. ABC. You can have ACB.
So now this is, let's say this is A dash here and this is B dash here. Okay. So these are two sequences. Let's say it's the same code and here this is not an intron. This is an axon now. So now the same DNA, same genetic code is going to produce a different protein because this is like let me grab the rubber.
Let's suppose that the RNA would be, because it's going to be shorter, so it's going to be just consisting of three segments, ABC.
Yeah.
So the mRNA ABC, that's going to be one possibility. So we can construct it in that way. The mRNA can be ABC, possible mRNA ABC.
Yeah.
So this is one form of splicing. But if the splicing enzyme does one like error over here, the splicing enzyme can splice them or stitch them together in an order BAC.
Yeah. So the main issue here is that, let's say what Substantia is talking about here is that, let's say it's ABC. So this let's take it there. So this is basically, this one is A, this one A, then this is B, and here this is C. And let's say the splicing enzyme has to join first this A, then this B, and then this C. So this is A, this is A. I am going sideways now. And this is C. So excuse my poor drawing, but what to do here? So let's say the splicing enzyme that is basically responsible for stitching these, because what happens is that the splicing enzyme would take the portion A, then the portion B, and then the portion C. And now it has to stitch these three together. And then this is the whole sequence. And ABC becomes an mRNA, and then it goes and encodes the protein. But let's say for some reason there's a mutation, and that could be due to a frame shift or due to something else. And now the splicing enzyme just forgets or makes a mistake. It joins A, and then instead of B, it joins C. And then after that, it joins B.
So as a consequence, a totally novel protein comes into being, a very different. So in order to get that ACB, you do not have to take the route of point mutations. So that Ghalib has illustrated very clearly now, that in order to get the because A in itself is not a single nucleotide. A in itself could be a sequence of ATCG, ATCG, things like C in itself is a sequence, B is in itself is a sequence. So in order to generate that sequence, effective sequence in RNA, from the original sequence, you do not have to take a strange route of point mutations. So suddenly you can start exploring many different variations in the landscape very quickly. And splicing enzyme is actually one of the ways we will talk about that. All these creationist people, one mistake that they do in their analysis is this, that they think that the mutation rate or the rate at which things are changing is constant. Okay, so
And
that,
secondly, yeah, please.
yeah, so we just saw that how the rate can be drastically increased, like a protein which probably never existed in the past, in evolutionary past, suddenly came into being because of the splicing enzyme, because of alternative splicing. And that can actually accelerate the rate of mutation, accelerate the rate of evolution as well. There are so many other ways and so many other mechanisms that change the speed or increase the speed of evolution. We will talk about epigenetics and other things. But even within the realm of genetics, this is one way, which is that you have got alternative splicing as a means of generating evolutionarily unprecedented proteins, brand new de novo proteins.
And secondly, these creationists, these intelligent design people, they always underestimate the robustness of the genetic mechanism that exists within the living organisms. This is just one example. There are other epigenetic factors that use the same genetic code to make different proteins. So if a promoter or an enhancer is behaving in a different way, it can even change the protein into other things. And same genes are used to do different things in different organisms, the same genes, because their epigenetic mechanisms are different, and that's why those proteins or those genes behave in a totally different way. So this is the robustness of this genetic mechanism that is within all of us that they totally underestimate. And that's basically the most wrong thing about thinking about evolution, because evolution is what is the outcome of the robustness or the change in the overall mechanism which creates the proteins. And that can happen on the genomic level, that can happen on the epigenetic level, that can happen within the splicing phase. It can happen on RNA and mRNA level, and it produces something novel, something new. And that is how organisms are changed. So that is why I was saying that I don't understand. These people are doing it deliberately or they don't understand it. Talking about Stephen Meyer, I have read two of his books, and I was totally unconvinced by those books, because whatever he wrote in those books, there is a genuine scientific explanation of all of those things. You can find those online, and if you develop a not very advanced understanding of evolution, you only develop a basic understanding of evolution, you at least understand how the mechanisms do work. So, Substantia, we are going to move on to the retroviruses now. Do you want to add something here before that, or should we move on?
No, you go ahead. You finish the idea of like this topic of endogenous retroviruses and transposable elements, and then I will start talking about epigenetics. So carry on.
Okay. So what are endogenous retroviruses? What are basically retroviruses? Retroviruses are basically packages of information, and that's not DNA information, that is RNA information. So what a retrovirus does is it enters your cell, and it is basically a genomic RNA. So this is an RNA. It does not have thymine, but it hijacks the machinery of the cell and it translates itself into DNA. And it hijacks the machinery of the cell, and then the cell starts to produce copies of that virus. So this is basically how a retrovirus works. So the main fundamental idea here is that whenever a retrovirus enters your body, it enters a cell, it copies its own genetic material, which is packaged in itself. It sends that genetic material into the living cell, and through the process, through the same process through which the cell manufactures proteins and different mechanisms for itself, the virus produces its own copy. So retrovirus insertion is basically a huge mutation, because there are viruses that are maybe tens of thousands or hundreds of thousands of base pairs long as well. There are huge viruses as well there, and we can think of it as maybe 1,000 base pairs inserted into your genetic information at once through a retroviral infection. And retroviral infections are very common. You and I, we had a lot of retroviral infections during our lifetimes. HIV is a retrovirus. There are a lot of other diseases in the world right now that people are suffering from. They're caused by retroviruses. So that's there. So there are thousands of endogenous retroviruses in human genome. So right now, endogenous means that a retrovirus, someone who goes into a place and makes it their home. So this is what retroviruses do. They go into a host and they transfer their information into the genome of that individual organism, and that information stays there forever. Through the process of reproduction of that organism itself, it also replicates the information that was inserted by a retrovirus at some point into the ancestor of that organism. Let's say my great grandfather, maybe a million years ago or 2 million years ago, was infected by a retrovirus, and that retroviral DNA or that retroviral information became part of their genome, and through sexual reproduction they gave it to all of their progeny, and today people have those things. So retrovirus insertion can have three effects on the host. First of all, they could be harmful, like HIV is. They could be neutral. The viral information will become part of your genome, but it wouldn't give you any kind of problems. But thirdly, they could also be beneficial. And there is clear evidence today, this thing has been studied a lot, that there are viral genome sequences that are basically were inserted by the viruses into our genome and genomes of millions of other organisms. But today they have been adopted there. They are now performing beneficial duties in those organisms. This is well studied. This is I wouldn't say this is totally new information. Maybe this was all discovered about 50 years ago. So this is how it happens. And secondly, retroviruses also tell us that we have evolved. And like we can see here, this is HERV. HERV. This retrovirus is found all over the human genome. And the main thing is that, as we say, we and chimps have a common ancestor. And among all these great apes, chimp, gorilla, orangutan, gibbon, and rhesus, like these are two other monkeys. So among all the great apes, chimps are the closest genetic relatives to the Homo sapiens or human beings. So, and gorilla is a little bit far than that, and then orangutan is. So what we see is that all these three genetic cousins, they have the same HERVW virus on the 205 same locations between chimps and humans. So what am I talking about? If this is a representation of the human genome, you can see that we have 23 chromosomes, and the 24th is the sex chromosome, and these are the pairs that we have. So if I only try to put this 205 times on the whole genome, this is what it would look like. So it's impossible for us and the chimp to have this retroviral insertion at the same points in our genome if we were not descended from the same ancestors. It would be impossible to have these same retroviral insertions on 207 same points with the gorillas, between the gorillas and the humans, if evolution did not happen. So this is proving a lot. These are very long mutations that it's impossible for these mutations to happen through point mutation. The only way that can happen is through retroviral insertions. And this tells us that maybe hundreds of thousands or millions of base pairs can be inserted into our DNA, and that becomes a permanent part of our DNA. And if genomic information can change in this way, so this also provides a very strong pathway for the development of new proteins as well, because in the end, what's happening? Even virus is creating new protein, it's creating proteins. And when it gives you the information that is able to create viral proteins within your cells, then it also gives you ability to create new proteins. So what David Gelernter was saying, that is totally wrong. He was saying that how come there's a possibility that a new protein is created, a novel protein comes into being in an organism. The organism does not need to create, or the organism does not have to come up with a way to create that new protein through point mutations, because it can just get that genetic information to create a new protein through viral genome as well. So this is what can happen. Even a viral protein that comes into an organism, and later on that organism copulates with other members of its species, and through reproduction that viral protein is going to spread. So that viral genome is going to spread into other organisms, and let's say the infection happened in a few hundred of those, the members of those that species. But when that is multiplied into maybe, let's say, 100,000 members down the road, maybe 10,000 years down the road, or maybe 1,000,000 years down the road, it has multiplied into 1,000,000 members. So then there's the chance that that same genome, that same gene, or that same ability to create a new protein can definitely come into, become useful through point mutations as well. So this is the chance that the biological mechanisms overall give you all the time through different pathways. So as we talked about the splicing and the stitching pathway through which different new proteins can be made, this is also a pathway through which different proteins, new novel proteins, can also be made. So this is, maybe we scientists now believe that there is a huge chance that a lot of our genome is basically the genetic information that has been brought to our genome in such a way. And if someone does not believe that such an information was inserted by viruses, so they can't be more wrong, because this has been proven right. It's absolutely true, because in the human genome there was a sequence that resembled, that scientists knew that it was maybe a viral sequence. It was inserted by a virus. So they reconstructed the same viral DNA, because it was inserted at many different points in the human genome. And what they did was, from the different point mutations and mutations that had happened within that sequence, that was basically inserted by a virus at a long lost point through in our human history, in our evolutionary history. So they reconstructed the functional copy of that virus, And when they provided it an opportunity, the virus that was only part of our genome now, it did not exist. It was extinct, maybe. And it had not lived, maybe, we don't know, whenever it was inserted into our genome. If it was inserted 2 million years ago, then after that time, this virus did not exist in the world. But when scientists reconstructed that genomic sequence, and it again became a virus. So these are actual electron microscope pictures of that virus. That was basically when it not only formed through the machinery of the cell, it also exited the cell in the same way that living viruses do today. So this is what has happened now. So we know that a virus that was dead for millions of years is today alive once again, when its genomic sequence that was highly mutated within the human cells, it was provided an opportunity to take its original form through the genetic engineering that scientists did. It again came into being. It became alive. So what was done? So they got the sequence of that virus. They packaged into the viral proteins, the viral covering, and they infected human cells with that virus. And then that virus again became a living virus. The scientists ironically named this virus Sleeping Beauty, that was sleeping in our genome for maybe millions of years, we don't know. And it again came to life. So this is like bringing a dead person back to life. This is how it is. So basically, when you deny evolution, you are actually denying the capability of genome of an organism to change and evolve on its own. So even this genetic information that we get through the viruses alters our genomic sequences, and we know that our genome is changing, and it's changing in big ways, like we explained two of those ways right now, and that's just huge. Our genome can change in huge ways. So it's not only this, there are a lot of other mechanisms. Point mutation is thinking of point mutation as the only mechanism of change within the genome. That's absurd. That's just totally ignorant, because I think we have only discovered so far maybe 100 ways in which genome can change itself. We don't know, even there could be millions of ways in which an organism's genome can change. So that's the main thing. Secondly, there's another thing. So we have different genes in our bodies, and these genes are responsible for making different proteins. And every time a cell divides, these genes are copied. And sometimes a gene that is copied once is, through some error in the copying mechanism, is also copied once more. There are rarely any genes in the human genome that do not have copies of those genes. Those copies that are made by mistake within the human genome, these are known as pseudogenes. Because when a pseudogene copy of a genome is made, what happens is only one of them remains active. But that also happens that both of them remain active, and they perform different functions, slightly different functions. So this is basically, the globin gene is basically responsible for making hemoglobin in warm-blooded in animals, basically. So there are two clusters of globin genes in humans. There are a total of 16 16 copies of globin gene on the alpha globin gene family on chromosome 16, and on the beta globin gene family there are 11 chromosomes. There are 7 on chromosome 16, the alpha globin gene family, and there are 6 on the beta globin gene family. So there are 13 copies of the same gene that makes hemoglobin in you and me. There are 13 copies, but only 3 of them are active after birth. 3 more are active when you are in your mother's womb, because that's a totally different, mechanism of getting oxygen from your mother's blood. So that requires a different set of genes when you are in your mother's womb. But 7 of those copies are pseudogenes copies because they don't do anything at any time of your life. They are just sitting there doing nothing. But 6 of them function, 3 of them function when you are in your mother's womb, 3 of them function when you and I came out of our mother's wombs. So how come these copies were made? So even by studying the alpha and beta globin gene clusters in different organisms, we can develop a tree of evolution of those organisms. Let's say we have 13 copies of those genes. Chimps would also have 13 copies or 12 copies. If we have 13, they have 12. This means that we are very close to them. Let's say gorilla has 9 of them. This means gorilla is a little bit far away. Our common ancestor with gorillas were a little bit more distant than we had with the chimps. This also proves how not only the genome is changing, how new functionality is being made, acquired from the same gene by small alterations in the gene. Because the gene can be, we will once again grab, Substantia, do you think we have time for this? Should we grab the tab once again?
Up to you. There are lots of things that we need to cover.
Okay, so I will just explain this, that let's say there is a gene and that gene is copied once more within the genome, and then the copy that was made, because it's not genetic, it's not active, it's not encoding into proteins, it keeps on mutating through point mutations or other frameworks of mutation, and maybe a million years down the road, what happens is it somehow becomes active with a new functionality, doing something that was totally not being done before. So Substantia, I think here we have explained different types of mutations.
Yeah. So what you are
These are just a few. There are maybe a dozen more that even I can explain to you guys.
What you are telling about is technically ways of having neutral mutations, because while that copy is not being transcribed and not being translated, it is not open to selection during that time. So it can keep evolving, and then it can get co-opted. So there was a guy who is credited with this idea of neutral evolution. His name was Shikimura, and that's called the neutral theory of evolution. And somehow many people present Shikimura's neutral theory of evolution as something like an adversary to Darwin. However, if you go back to Darwin's Origin of Species, Darwin is absolutely clear by telling that there are certain kinds of variations which are going to be beneficial, which are going to be harmful, and some of them are going to be neither harmful nor beneficial, which means neutral, and they can later on be co-opted. This is exactly what Darwin says. So in any case, so let's see what has happened so far. What Ghalib has explained so far is that the calculation approach of Gelernter, or even the attempt to begin to start calculating probabilities, is basically a very myopic approach, even in the framework of point mutations. We showed you synonymous mutations, synonymous codons, synonymous protein structures, so on and so forth. So that calculation was flawed. Then Ghalib told us about different other mechanisms which go beyond point mutation. We talked about frame shift. We talked about alternative splicing. We talked about these retroviruses being integrated into the human genome through something which is referred to as the jumping activity, or like the transposable activity of these retrovirus genome into human genome. So there are so many variations on top of point mutations. Now, I think we can spend some time on another mode of variation inheritance, which is referred to as epigenetics. We have talked about epigenetics before, but let's start with an example of epigenetic variation, and then we can quickly go through certain papers that talk about the role of epigenetics as a parallel mechanism that controls evolution. Let this be very clear, guys, that if you are ultimately committed to something like a gene-centric view, you can be wrong. And for 40 years almost, that while the gene-centric view and considering the gene as the unit of selection in evolution, while that is a very, very beneficial and useful, like a pedagogical aid, because it helps us in understanding, but epigenetics has been known to us for a very long period of time. The moment we acknowledge that there is epigenetic inheritance and a parallel mode of creating variation, and a variation that can be inherited over multiple generations, of course we are agreeing that the selection unit can be multiple. The selection can be at multiple levels. It can be at the level of epigenetics, it can be at the level of genetics, it can be at the level of an organism, it can be at the level of the microbiome in the inside the organism. So there can be multiple levels of selection going on. Point is, all of these parallel mechanisms, all of these parallel units of selection, so to say, all of them are actually strong arguments in favor of evolution. The more variation you have, the more you prove that this variation is heritable, the more you prove that these variations are associated with differences in fitness. They like confer differential fitness to you. You are making Darwin's argument. So if someone says to you, you know what, like the neo-Darwinism, which was gene-centric, is dead, you know what, in the demise of that kind of neo-Darwinism lies the resurrection of the original Darwinism, because Darwin at the time subscribed to the inheritance of acquired characteristics. And you know what, however strange it might sound to you, it doesn't sound strange to me, because I have known about epigenetics for the last how many years. Like I wasn't born last Friday. So in any case, so let's start with epigenetics, because through epigenetics mechanisms, we now know beyond reasonable doubt that acquired characteristics can be passed on to next generations. Not only next generation, not 2, not 3, we have got evidence up until like 20 generations of acquired characteristics being passed like down the generations for about 20 generations. I will talk about that. Let's start with paper number 6, which is about epigenetics.
Yeah, I'll just bring it up.
While Ghalib brings up that paper, so for English audience, I am hoping that we have got an overlap in the audience from the other channel, because on the other channel we do talk about epigenetics a lot. So epigenetics, guys, is just a set of mechanisms which can actually alter the expressions of genes, because at the end of the day, it's the expression of genes that matters. So it can alter and change the expression of genes without actually altering or changing the underlying DNA sequences. So there can be switching mechanisms or regulatory mechanisms that can switch on or off a gene. There are many different mechanisms. I will not get into the details. The DNA methylation can switch off certain genes. Acetylation can switch on. There are modifications in histone. There's non-coding RNA, so on and so forth. So this is an interesting study just to get things a little bit stirred up, that what's the potential of acquired characteristics. So here, if you read the title of the paper, they say that parental olfactory experience influences behavior and neural structure in subsequent generations. So what it actually means is that a mouse, if the mouse was exposed to a certain kind of an odor, so parental olfactory experience, so that can actually demethylate certain genes, okay, which means that it can switch them on, and the mouse can be conditioned to be fearful of that odor, okay, as a result of demethylation of a particular gene. So if I will read out what that's called the OLFR151 gene. And then, oddly enough, what happens is if you analyze the sperm of that mouse, the gene in that sperm is also demethylated. As a result, this characteristic of being afraid of that odor is passed on to the next generation. The offspring is also behaving the same way. It's an acquired characteristic. So you see, parental olfactory experiences, like the experience in the lifetime of that mouse, that induces a change that is then passed on to the next generation. So what is it saying? The highlighted part, that the bisulfite sequencing of sperm DNA from the conditioned F0 males, okay, and F1 naive offspring revealed CpG hypomethylation, like less methylation, in the OLFR151 gene. So not only in the father, but the offspring also has got the same methylation characteristics, which means that the epigenetic change, first of all, the epigenetic change was induced by an experience in the parent, and then the same epigenetic change got inherited. So, and what it is saying is that this F2 inheritance and cross-fostering revealed that these transgenerational effects are inherited via parental gametes. Welcome back to Lamarckism. Welcome back. Okay, so this is what precisely Lamarckism is. So it is now beyond reasonable doubt, and this is a parallel process of variation that can be inherited, that can have differential fitness, that can be passed from one generation to another generation. And anyone who has studied evolution, this is raw material, this is the fuel for evolution. Okay, over to paper number 7.
Just give me a second. I will go to paper number 7. So this is paper number 7. Yeah.
So paper number 7 is going to be, we will very, very quickly skim through this paper. So this is basically the inheritance of epigenetic variations in both plants and animals. Okay. So so this is the review paper, guys. And if you read this highlighted part, this review is based on 500 studies, over 500 studies. So they're saying that we systematically reviewed over 500 studies. I'm reading the yellow part from the past.
Substantia, can you just in one line explain to the people what is the review paper? Because many people won't know.
Yeah. So if you are submitting the results of a particular research that you have conducted, so you would say that that's a research article or an original research article. If you are submitting a review article, that actually means that you have systematically reviewed many studies, and you have looked at studies from, let's say, last 10 years, 5 years, And the gist or the crux or the central message of the body of a literature in a particular field, that is what you are communicating. Not only that, you are also communicating some of the pros of the research directions that have been taken in the past, some of the issues, some gaps in research, and a good review paper also in the end concludes with giving some directions for future. It's not just a aggregate of what has already happened. A good review paper is kind of like a signpost as well, that you know what, this is what has happened. This is something that needs to happen. These are the current deficiencies. So that's what a review paper is. So it is kind of like a gist or the crux of research in a certain domain. So what they're saying, that they conducted a review in which they reviewed over 500 studies, and they identified trends in the frequency of the epigenetic inheritance, that how frequent it is, and then of course whether it is a function of reproductive mode, and whether it's during the germline development, and whether it affects the germline or not. Okay, and what their result is that there is multi-generational persistence, because in order for evolution to work, not only there should be transmission, but there should be tendency for that variation to persist over multiple generations. So there is evidence that there is multi-generational persistence of epigenomic patterns. So some of the biologists say, you know what, epigenetic changes can go up until 2-3 generations, so evolution cannot work on it. No, that's not the case. Epigenetic repeatedly has been shown to go much beyond multiple generations, and there is strong evidence that epigenetic has played key role in like major evolutionary steps, especially in the case of like sudden and fast and high-paced evolution. Okay, if we go to page number 2, and this is addressed to, again, students of biology or anybody. Actually, we are addressing this to those 3 gentlemen as well. So go to page number 2.
This is
Top of
this
page
is
number 2.
page number 2. Top of page number 2. This is page number 2, right?
No. So
okay. Actually, I don't know why this is not showing. Yeah, this is page number 2.
This is page number 2. So I purposefully highlighted this part. So any biology student who has taken biology in high school knows something Weismann barrier. Once upon a time, like Weismann was the anti-Lamarckian guy, and the Weismann barrier was this: that if you have got any changes acquired during the lifetime, they cannot be passed on to the germline. So what this is saying is that while it was once accepted that the Weismann barrier prevented somatic cells from altering the germline after cell differentiation, this idea has been disproven through research on epigenetic inheritance. Okay, so goodbye to Mr. Weismann and welcome back to Lamarck. I'm dramatizing on purpose. Okay.
Yeah.
Okay.
Once again,
If you go to page number 6, let's go to page number 6.
page number 6.
So that's
5 and this is 6.
Yeah. I want everyone to look at this like chart.
Should I zoom out a little?
Yes, actually zoom out. Yes. So what it is showing is that as a mode of DNA, like epigenetic variation, as I mentioned, there can be multiple modes of epigenetic variation, like DNA methylation, histone modification, ncRNA at the bottom, like non-coding RNA. This is an excellent depiction based on these studies. What it is telling is, so the x-axis is kind of time, or if you look at the top of this box, there is this F0, F1, F2, F3 up until F20. So these are generations. So the title of this graphic is, of course, epigenetic persistence across generations. So what they have shown is that there are certain kinds of changes in certain species which keep persisting. For instance, if you can scroll down and maybe now zoom in, so the highlighted part in the caption of this figure is this like one study on histone modifications of this this particular species
It goes in.
of histone modifications that lasted for from generations F1 to F20. The histone modifications actually persisted still from F1 to F20. So one needs to just educate oneself with what has been happening in the last 40 years. Again, Stephen Meyer was saying that the evolution was perhaps an excellent theory proposed by Darwin in the context of 19th century knowledge, but come 20th and 21st century, we have got molecular biology and whatnot, and therefore it is outdated. Okay, you know what? In 19th century, the understanding was actually the correct understanding as vis-a-vis the inheritance of acquired characteristic. That 19th century understanding was correct. We know we spent the entire century, like good part of 20th century, considering that, and making fun of people who would say that the Weismann barrier can be broken. But the lesson is that 19th century understanding was correct and the 20th century understanding was wrong. Okay, we have got reasonable, beyond reasonable doubt evidence for epigenetics. Now let's go to paper number 8. I think this is a comprehensive paper which documents the role of epigenetics, transposable elements, sudden fast changes, punctuated equilibrium. Some creationists are fond of talking about punctuated equilibrium, thinking that maybe punctuated equilibrium is an objection to evolution. Actually, punctuated equilibrium is part of the story of evolution, and it is actually enabled by epigenetics. So for those people who don't know what punctuated equilibrium is, the idea of punctuated equilibrium as proposed by Stephen Jay Gould and Niles Eldredge was that for many long periods, there is not much evolutionary change, and that's called periods of stasis or no change. Okay, and then there are sudden bursts, like suddenly organisms evolve very rapidly. So therefore, what they coined the word of punctuated equilibrium. The normally is an equilibrium, but this is punctuated by sudden bursts of like very fast and rapid evolutionary change. And the fossil record also speaks of this, and the evolutionary record, the genetic record also speaks of this. However, historically, people used to say, you know what, the whole idea of evolution is that small changes accrue and accumulate over a long time, and then given enough time, you will have that change. So how do you reconcile punctuated equilibrium with molecular biology, understanding of evolutionary change? Precisely because of molecular biology, we have got like satisfying mechanisms that can potentially enable and fuel punctuated equilibrium. By the way, that the thing that we just discussed a few minutes ago regarding splicing, alternative splicing, if you have got too many different variations of splicing being tried out, it can cause sudden bursts, and usually you have stasis and no change. In any case, what's the title of this paper? So this paper's title is Transposable Elements and an Epigenetic Basis for Punctuated Equilibria. Okay, I think the title says a lot, but I think if Ghalib you allow, I want to actually cover this paper in more detail.
Yeah. So Substantia, what we can do is that we can transfer some of the other things to the next stream. Because I think we are 1 hour and 39 minutes into it. And I don't know how much time you have. So we can cover this one today. And maybe in the next stream, we continue with this. Because I think we are going into taking a deep dive into all of it.
Yeah, I think we will need at least 2 episodes to fully address it. So let's cover this then.
Yeah, sure.
So here I'll just, I mean, I've explained this, but I think in order to do justice to the paper, let's read these sentences. So what they're saying is that evolution is frequently concentrated in bursts of rapid morphological change and speciation followed by long-term stasis. Okay, so this is precisely what punctuated equilibrium is, that for very long periods, no change or very little change, but then there's sudden change. Okay, and they see that this pattern of punctuated equilibria results from an evolutionary tug of war between host genomes and transposable elements mediated through the epigenome. So the current understanding that is developing is this, that these sudden changes are a product of an interplay between the activity of transposable elements and epigenetics. Okay, so what happens is that the transposable, actually, transposable elements, as Ghalib was suggesting, so that they are also referred to as jumping genes. And we have talked in other streams about how they were first discovered by in corn, that in response to stress, like Barbara McClintock's work, who got the Nobel Prize in 1983.
We've
for
talked
this work.
about her a lot on our
Yeah,
Urdu channel.
Yeah. So those are the transposable elements, or those elements or parts of DNA which jump to the coding part of the DNA as a result, leading to alternative, like, expression profiles and alternative proteins, or maybe total junk as well. One of the best explanations of how epigenetic mechanisms evolved is that epigenetics actually evolved in response to, as the defense mechanisms against transposable elements, to switch off the activity of the transposed elements in the coding part. Okay, so this is what they're saying.
And transposable elements themselves, maybe you should say a few words about that as well, because
and transposable elements. So what was your question?
So my question is basically, how did transposable elements come, and where did they come from? Secondly, we should also explain a little bit about the transposable elements, because maybe some of the people do not know what we mean by transposable elements here.
Yeah, more often than not, they are invaders. They are, like, technically, you might want to call them as parasites or symbionts. Like, they are from retroviruses are transposable elements. Like in those parts that correspond to retrovirus DNA, the transposable activity is actually extremely elevated. So they come from other organisms, and they have just found a good home in like a host genome. You can imagine that. So there are parasites, but then they get integrated into it, and as you will see with epigenetic mechanisms, they actually help in the evolutionary process and co-evolve. Okay, so that's the understanding, that the epigenetic regulatory mechanisms, they maintain stasis, which means resisting the change by suppressing the movement of transposable elements. However, under conditions of stress, what happens is that, or like they're saying, that climate change or invasion of new habitats, the epigenetic regulation is disrupted. The methylation patterns, for instance, could be disrupted, and that is the time when transposable elements go all hell
totally go haywire.
the all hell loses, and they can actually start having expression-based changes. Of course, they can get themselves inserted in different parts of the DNA, but if the corresponding epigenetic mechanisms are switching off those parts, it's basically neutral change. However, when the epigenetics switching mechanism is compromised under stress, then transposable elements can actually express themselves. Okay, so if you look at the right side, they are suggesting that the growing evidence indicates that epigenetic regulation evolved to suppress these transposable elements. So this is on the right column. So the epigenetic regulation evolved to suppress these transposable elements, and basically transposable elements are parasitic sequences, exactly what I was saying. It's a parasitic sequences that comprise a large fraction of other eukaryotic genomes as well. Okay, so let's move to page number 2.
Okay, column 1 or column 2?
Yeah, column 1. So what they are going to do is they are going to present hypothesis, like
Right,
evidence. No, that's fine. They are going to present evidence in support of this hypothesis. It is still referred to as hypothesis, but although there is so much evidentiary support, but I mean, who cares about the labels? So it's telling that the support for this hypothesis, which is referred to as the epi-transposon hypothesis, actually it comes from multiple lines. Okay, it comes from genome studies of comparative genomics, like molecular biology, molecular systematics, environmental epigenetics, and also radiation research. So there are two parts to it, two main parts to it. That the transposable elements, they are one of the key driving forces in the silencing of in the evolution of the epigenetic silencing mechanism. So they are also epigenetic mechanisms are also referred to as silencing mechanisms because the effect of methylation per se is to switch off the gene or silence the gene. But there are other epigenetic changes as well, which can increase the activity of the gene as well. But precisely because transposable elements had to be switched off, so therefore the discussion is in the context of epigenetic silencing. And secondly, transposable elements in and of themselves have been a rich source of evolutionary and genetic diversity in novel genes. Okay, and this epigenetic silencing or repression of transposable elements that actually breaks down in response to stresses or climate change, sudden change. So I think we can now jump to page number 4 and zoom out a little bit so that I know where we are.
So here, I would zoom out in this portion.
Yeah. So now so this is like transposable elements as a source of new genes and new like regulatory networks. So this is usually referred to as molecular domestication. So just like I was saying that initially you might want to say that transposable elements are like parasites, but when they start contributing to the genetic diversity of the host genome and start like affecting it positively, that it enhances the evolutionary fitness, you might want to say that you have domesticated that parasite. And that parasite is basically a sequence. So this is usually referred to as molecular domestication. And these transposable elements have one of the leading sources of genetic innovation. They talk about where does the genetic innovation come from? Okay, you have got transposable elements. And even the evolution of like the mammalian placenta, if you look at it and dig into the references, we will not dig into any further references. This paper is like deep enough as well. But the evolution of mammalian placenta is a textbook example of domestication of transposable elements, and you can read the references of this paper if you are interested. Let's move to page number 5. Zoom out and page number 5.
Yeah, so this is transposons unleashed.
Stress-induced breakdown and epigenetic silencing. So of course, as I mentioned before, that when the epigenetic mechanisms break down, that's the time when transposons start expressing themselves, because before that, if they have been able to insert themselves into coding regions of the host genome, the epigenetics would silence them. But when epigenetic mechanisms are compromised, that's when you have got transposable activity and expression increased. And these are the bursts. These are the sudden changes. So in punctuated equilibrium, when epigenetic mechanisms are compromised, that is when you have got sudden bursts of evolutionary change in punctuated equilibrium. If you go to page number 7,
Yeah.
so there's a punctuated equilibria and transposable element mobilization and expansion. So when the epigenetic mechanisms are working correctly, so you might want to think that then the jumping activity of transposable elements is not under selection. So they can keep expanding and they can keep moving. So that's the phase. The stasis phase, the long stasis in the punctuated equilibrium, interestingly, is also the phase when the TEs, the transposable elements, are actually moving around, and they are expanding as well. But they are not weeded out because they do not have any cost. Like the selection is not operating on them. Why? Because they are just switched off through epigenetic mechanisms. So that's the reason that during the equilibrium part, the stasis part, the transposable elements, they actually keep expanding. And one of the one of the expansion categories is also referred to as sign expansion, which is a short for like short interspersed nuclear element expansion. And if you, Ghalib, you are familiar with ALU sequences, right?
Yeah, definitely.
So,
Definitely.
so if you look at,
I have a
reference-
few slides about that as well, if
Yeah,
you want. Or rather, next time we can show that
definitely
about the
So-
ALU and those.
So
yeah.
if you look at this green highlighted sentence, what it says is that the comparative analysis of ALU sequences actually does indicate that all major like divisions or bifurcations that have happened in primates, they actually coincide with the moments, with the timeline of these short interspersed nuclear element expansion, which means that ALU sequences diverge when you have got expansions of these transposable elements, and they are responsible for those certain bursts. And if we go to page 9 and just read the conclusion.
You want to read from here? Or, yeah, okay, this is part of the
Is this
conclusion.
page 9?
Yeah, this is page 9, and this is part of the conclusion. Basically,
Maybe the left
conclusion
column.
is at the bottom of column 1.
The left column. No, the left column. Column 1.
Okay. Okay. Yeah. Okay. There's this.
Yeah. So what they have actually shown in this paper is this, that the strength of this epi-transposon so-called hypothesis, because I think the evidence and support is overwhelming. So the strength of this epi-transposon hypothesis lies in the ability to explain both long-term stasis and rapid diversification by providing an epigenetic link between environmental change and transposable element-driven genomic innovation. So the strength of a theory lies in the range of the phenomena that it can explain, right? So this is what they are stating, that one of the biggest objections was that, you know what, maybe molecular biology was kind of a challenge to the fossil record and other phylogenetic record that speaks of that is consistent with punctuated equilibrium. The power of this interaction between epigenetics and transposable elements theory or hypothesis, I think it is very close to being elevated to a theory. So the power of this hypothesis is this, that they can explain both long-term stasis, very slow evolutionary change, slow, like more like a little bit change. Like, I mean, just think about it. I always say that, you know what, life was stuck at bacterial stage for 2.5 billion years. Okay, so because there was the evolution was so slow, and there were so many failed attempts in trial and error, because these mechanisms were themselves evolving. And then, so life got stuck in that bacterial stage for over 2.5 billion years. But then the entire flourishing of multicellular life almost happened in like last 600 or 700 million years. Okay, so interestingly, this epitransposon hypothesis explains both very slow evolution and sudden evolution. So that's the power. And I think, as we shall see, that epigenetics is one of the key players in, you name a thing, and you will find out that epigenetic mechanisms are at the heart of, at the center of that mechanism. If Ghalib, if you want to show one of the books by that author, Nelson.
Just let me bring it up. Where did I put it? Yeah, on the bottom of that. Or did I miss it somewhere? Where did I put it? Yeah, okay, now I got it.
So they talk about,
this
so
is
we,
the
we,
book.
so
This
they
is the
talk
book,
about
and
sudden change.
yeah,
We do
yeah.
not see
So
your screen yet.
yeah, now it's up.
So highly recommended by this author, Nelson Cabet. Like it's the epigenetic
I'm going to
mechanisms
read this
of,
one.
yeah, epigenetic mechanisms of the Cambrian explosion. So they talk about sudden change, how like about 540 million years ago, like within a span of like around like a very small period of time on an evolutionary scale, you suddenly see a flourishing of things. So this is the result of the interplay between transposable elements and epigenetics. And totally, so epigenetics has got this power of explaining Cambrian explosion as well. Okay, I know that Cambrian explosion is, there are some classical explanations of Cambrian explosion as well, and there are fossils like stromatolites have been discovered before Cambrian explosion as well. So the previous objections are kind of like outdated and redundant. In any case, still, it is true that there is this sudden flourishing of life, and epigenetic mechanisms can explain, because they can explain variable rate of evolution. Okay, they talk about like development of body plans. There are epigenetic mechanisms behind the early development and development of body plans. We will get to do all those things. They talk about speciation. Yes, we will address those questions. There are epigenetic mechanisms behind speciation as well. So you just be willing to learn. Dr. Gelernter,
Yeah.
Dr. Berlinski, Stephen Meyer. Honestly, I mean, we will continue in episode number 2. And I, as an ordinary
We are going to do that next Friday.
and we are not speaking, to be very clear, we are not speaking from a position of authority. I am speaking in the capacity of a lifelong, ordinary student of these subjects. And I think anybody can invite you to like learn more, of course. And I think if you approach these disciplines with an open mind, and what's the next thing? Open mind is not enough. Open mind and hard work, Dr. Gelernter. Respectfully, you will have to do the hard work because you are a computer scientist. So you will have to do all the hard work to learn these things. But I have no doubt that you are in a much better position. You are a smart person. And it appeared to me, I'll give you the benefit of the doubt, that your position was quite sincere. I'll give you that. And precisely because of that, I'm addressing only you, that if this message gets to you, be open to all these different mechanisms, and then be my guest to get inspired by Stephen Meyer's book, okay, which is
Yeah.
honestly
that's,
a joke.
I
which
don't
is
know
a-
how that would be inspirational. Yeah. That's,
yeah,
that's a big question mark for me.
which is a joke. But in any case, we are just scratching the surface today. We will continue in episode number 2. We will address all those questions that you raised one by one. I think we have laid the foundations in this episode that there are alternative sources of variation, okay, and alternative engines of evolution. Gene centric view is not the end game, okay. I don't know if you growing up you were a fan of maybe selfish gene, that's why you were so committed endlessly to the idea of gene being the unit of selection. There are other parallel units of selection, and all of those discoveries in the last 30-40 years have been strengthening the case of natural selection and evolution. So I think I'm going to stop. I took a long time, but I think we can continue next time. We've got lots of material to cover.
I think we don't even know that it's just going to be the next episode that's going to, conclude this topic, because once we started discussing it, even I realized that, the depth of this topic is not what we estimated. It's way more deep than that. So thank you, Substantia, for all your valuable input. We are going to keep bringing up evolution. As I told you, it has been one of the major themes of our Urdu channel as well, and more so for this one as well, because we want to make this one way like education is the main emphasis of this channel, and also the other one, but that's a little bit more interactive than this one. Here we want to keep things precise and uh, more focused. And to be very frank, we wouldn't have time to expand on this channel in the same way as we have on the other channel. So that's also a limitation that we are facing right now. And hopefully you liked this stream and the discussion between the two of us. Hopefully you learned something from it. You can always give us feedback on Ghalib@ghalibkamal.com. That's basically my email. That is GHALIB @Ghalib Kamal. That is GHALIBKAMAL.com. And you can send us a feedback there. You can always send us feedback in the comment section. We did not read the super chats, but we really appreciate the people who support this channel. And because I did not want to break the things how they were going, how you were explaining, so that's why we are reading them at the end. So Shoshin said, Ghalib, you can easily beat all my professors. Thank you very much, Shoshin, that you think highly of me. And Substantia is 1,000 times better than all the professors that I had. And then Secular Humanist said that love your work. I rarely see a challenge to evolution in general, but Muslims contest that humans evolved the same way as other animals. Is there a separate mechanism of evolution for humans? No, there isn't. We evolved through the same mechanism. It has been a topic of discussion on the Urdu channel, and we would discuss human evolution also in a lot of depth in the coming episodes. So you subscribe and say, and turn on all notifications that you know when we are going to do that. Human evolution is going to be discussed at length on this channel, and we have plans to discuss it deeply, and we are going to cover it from different angles. Epigenetics is one of those, genetics is another one, fossil record is also another one. So it's going to be an expanded review of the things that we've already discussed on the Urdu channel on this channel as well. So Substantia, any closing remarks before we go? Guys, this is not over yet. It's going to be concluded hopefully in the next episode, next Friday. So
Yeah,
turn on
I
the
think we will.
notifications.
Yeah, if someone
Yeah.
has been steadfast up until this point and is watching, so of course, when we will start next episode, we will give a 5-minute review or like 10 minutes review of what we covered today, and then
Mhm.
carry on from that point on, because there's still a lot coming. We want to present a mathematical challenge to the deniers of evolution in the form of a mathematical proof. We also want to explain that even within the scope of point mutations, how some of the empirical evidence suggests that under certain conditions, the rate of beneficial mutations, the proportion of beneficial mutations actually surpasses the proportion of harmful mutations. So that contention that beneficial mutations are rare and harmful mutations are more, that is challenged. And that is empirical results. So that's what the empirical results show. We have covered those things in the Urdu channel. And yeah, so then we will talk about, again, point you to some references, how epigenetics can be used to explain Cambrian explosion, what's the role of like genetics in speciation, and what's the genetic advantage of speciation. We will question all the classical details. So last 40 years, biology has advanced. Unfortunately, the objections from the creationists, they are grounded in older like biology. The cutting edge story that is being told, be it be the challenging the position of gene-centric view, whether gene is the unit of selection or not, whether the organism, the microbiome, as I say, or epigenetic inheritance, transgenerational generation inheritance, is that the unit of selection? All these questions, although they are questioning some of the accepted ideas in biology, interestingly, they are aiding the idea of evolution. So I'll conclude with that, and I'll see you next Friday.
Okay, thank you very much for watching us, and hopefully we'll see you next time. Thanks for the super chats that you guys sent, and keep supporting the channel, and we are going to see you on next Friday. Thank you very much, and if you like this and you learned from today's stream, do like and drop a comment that this video gets engagement, and hopefully we are going to do the next stream on Friday. And thank you very much for watching. We'll see you on the next Friday. Good night.
Good night. Take care.